These are photographs taken through the microscope, Isaac, of 3 different nuclei from 3 different persons' body cells. Okay? So, like, this is the nucleus from one person. Cell membrane has been washed away, cytoplasm's been washed away. That's just the nucleus. Here's the second person's nucleus, and here's a third person's. And, raise your hand when you know the answer. Wait, what's that? Raise your hand when you know the answer.
What? Oh, it's a scrunched up Y chromosome.
What?
I mean X chromosome.
Excellent, a scrunched up X chromosome. Inactivated X, right? Bar body. Okay, so what kind of I'm just going to point to a volunteer. Feel in order. Wait, do you guys like it when your teacher just like points to volunteers?
Yes. I like a box with their names on top.
Like a
yeah. I like when you like draw names. It's more fair.
Yes.
You want me to draw names out of a box?
I think it's more fair. No.
Raise your hand if you would like from henceforth for me to get a box, put names. Really?
It's a lot more fair than you just pointing us.
Well, I do that with the 9th graders because they're younger.
I think it's fun. It's like third grade.
Except make it like the box of fun.
Okay, we'll do that. That means I gotta get more of those popsicle sticks.
Just eat.
I get them at Walmart. Okay. All right, I want to know, I'm going to point to a volunteer, a victim, I mean. Tell me what kind of sex chromosomes the person would have who gave us this nucleus here. What kind of sex chromosomes would this person have? I'll point to Isaac.
XY?
Probably XY. Good. How about this person? Chloe? XX. Could this be anything else?
It could be XXY.
Right, could be a guy who's XXY, right? Yeah.
Or XXYY.
Yeah, that's possible too. Yeah,
Yeah,
that's possible too. What you're going to find out, you guys, as the year goes on, what you're going to find out in genetics, in human genetics, that is that anything that's conceivable is conceivable. Okay. How about this one?
XXX.
Could be XXX. Could be XXX.
Yeah.
Okay.
XXY.
Stand by. We're not doing that.
Oh.
All right. All right, now there's some vocab words I want to look at here before I turn you loose on the computer tutorial again. We're looking at page 6. A couple of vocab words and some root and stem words we need to pick apart. Aneuploidy. Aneuploidy. First off, there's a word here, ploid. Anybody know what ploid means? Maybe not.
Let out.
No. In genetics, it refers to a set of chromosomes. Ploid. So you've got room to write some notes down in here. So put like ploid and then a set of chromosomes. Ploid means a set of chromosomes. Let me show you the human ploid. The human set of chromosomes. There it is. That's it. Now that's a photocopy of a photocopy of a photocopy of an actual photograph taken through the microscope. No, wait a minute. How many? Photocopy of a photocopy. Many copies.
Can you just give us the original?
I don't have the original anymore. Here's another way of looking at the ploid. You've got a chromosome number 1 that looks like that, and number 2 that looks like that. You've seen this before, right?
Yeah.
There's your ploid. The ploid Look at that. Now that's only one set of chromosomes there. So sometimes we call this situation monoploid. Mono, oh, what's that prefix mean?
One.
Yeah, monoploid. One ploid. And we use in genetics a lowercase n to represent that. So n means the ploid number, okay? Which in humans is 23. What kind of cells would you expect to find that would contain, that would be monoploid, that would have,
Sex cells.
excellent, 23 chromosomes, sex cells? Sex cells are monoploid. Now sometimes we use another word, put that in parentheses down here. Haploid means the same thing. Half the number. Yeah. At least half the number that a body cell. Okay, back to page 6. That's what ploid means, a set of chromosomes. Now, take a look at this. I'm going to put something else up here. There's 2 sets of N. So we call that 2N. Diploid, good. So here we're looking at a diploid situation where we've got 2 sets of 23. 2 1s, 2 2s, 2 3s, and everything. Diploid. Diploid. What's DI mean? 2. Diploid. What kind of cells in your body are diploid?
Everything else. Everything's left.
Right. Everything except the sex cells, right? Bone cells, muscle cells, skin cells, pancreas cells, spleen cells, stomach cells, heart muscle cells, brain cells, all the other cells in your body. Diploid. Okay. Now back to page 6. All right. Does anybody know, I don't know what kind of etymology you guys have had, what the prefix EU means? What?
Say Europe.
Not Europe, no. EU.
Like eugenics is to find the best possible gene. So it would be new or improved.
Close. It means good or true. EU means good or true. Gabby, did you know that? Oh, you're smiling like you knew that. It means good or true. Isaac?
So when you have like eukaryotic cell, it's like a cell.
It means a cell with a true nucleus. A true nucleus, yeah. So EU means you or good. True or good.
What about like a eulogy?
That's where you're saying something good about something after they've died.
That's good.
Yeah. Euro?
You're the best.
All right, EU means good or true. So, when you're talking genetics, you guys, when you're talking genetics, look at this word up here. Let's just take part of this. Euploidy means true multiples of N. Any cell that contains true multiples of the ploid, so if you're talking humans, any cell that contains true multiples of 23 is considered a euploid condition. So any number times 23, we call that euploid. So let me ask you this. Look at this. Here are the chromosomes inside the nucleus of a skin cell. Is this a euploid condition? Bless you. It's diploid. Is that a euploid condition? Yeah, because it's some number times n. It's a multiple of n. It's 2n. How about if you're looking inside the head of a sperm cell and you've got 23 chromosomes here? It's monoploid. Is that a euploid condition? Yep, true multiple of n. Happens to be 1 times n. Now there are some plants. There are some plants that have like 5 sets of n, or 6 or 7 or 8 sets of n. And we call those polyploid. Have you ever heard that word, polyploid? Now the thing about plants is when they produce their fruits, if they're polyploid, they make bigger fruits than normal, which is kind of helpful. If you've ever seen ginormous, in the grocery store, ginormous apples, big ones, or strawberries that are like big as your fist. Well, not that big, I'm exaggerating. Big, big. They're probably polyploid. Because plant breeders, those people who produce new varieties of plants through plant breeding, have figured out how to create food crops, certain food crops that are polyploid, and the fruits that they make are bigger than normal. More desirable for people who buy those kinds of things. So, in plants, being polyploid is a big advantage. Now in humans, you're going to find out when you go to the computer lesson that if a baby is polyploid, like 3N, that's called triploid, can't survive. It's a lethal condition. How would you get a human fertilized egg with 3 sets of chromosomes? Possible.
When they split up, like, you're going to need 23 non-disjunction chromosomes.
There's something that's more possible than even that.
Oh, 2 sperm both get in, but they're from the egg.
Here. 2 sperm cells that make a photo finish, and they both get into the egg at exactly the same instant. Actually, is that what you were going to say?
Or
They both get there in at exactly the same time. So you get 23 chromosomes from sperm A, 23 from sperm B, 23 from the egg, 3 sets of chromosomes, triploid, can't survive. Isaac?
is it possible for the outside of the egg not to harden by means of like a genetic process?
It's possible. And then you get multiple sperms going in. If
Does
that fertilized
happen
egg,
or no?
it can. That fertilized egg would die within a day, probably. It can't survive. So that would be polyploidy. That would be a polyploid condition, or that would be polyploidy. Let's talk about that fertilized egg that's got 3 sets of chromosomes, triploid. Is that a euploid condition? That's a little tricky. It is considered euploid because it's a true multiple of n, 3n. You with me? Now it's not normal, but it's euploid. I'm just bringing these words up and these root words up because you'll run across these in genetics, okay? So, what happens to a word if you put an on the front of it?
Not.
Right, it means not. So like, here's an example. Aneuploidy. Aneuploidy. Not true multiples of n. Aneuploidy. That would be not true multiples. And here's an example. Let's say you have a baby that's born with 47 chromosomes. That's not a true multiple of 23, right? That would be 2n plus 1 extra chromosome. We call that trisomy because you've got an extra, you've got 3 of one particular chromosome. That's an example of aneuploidy. Actually, 2 examples of aneuploidy, trisomy and monosomy. So if a baby has 321s, Down syndrome, then baby does have a multiple of 23. It's got 46 chromosomes, but one extra, 47. That makes it aneuploid. All right. This is like exciting stuff on a Friday afternoon, right? Are you getting these words, though? It's important we talk about those. Okay. Monosomy, the other kind of aneuploidy. You know how many trisomy 21 babies are going to be born in Indiana this year? Probably about 350.
Pretty close. Seems like a
In Indiana. Seems like what?
low number.
It could be up to closer to 500 now.
Does it go up or does it go down or something?
It's going up year to year because the population's increasing.
So if you had an area in which like you had a certain population of babies with Down syndrome on the board, if people kept like mating in that area, would that increase in terms of gene pool?
That's a good question. No.
No, would just stay constant.
Yeah. Because chromosome problems like Down syndrome don't run in families like gene problems do.
Oh.
When a baby is born with something like Down syndrome, trisomy 21, that's just a random mistake in meiosis, either in the formation of the sperm or the egg that made that child. A random mistake, chromosomes forgetting to pull apart during meiosis like they're supposed to, that can happen to anybody, anytime. And that's what we're going to look at down here. It's called nondisjunction, failure of chromosomes to separate during meiosis. It's called nondisjunction. That's a mistake. It's just a random mistake in meiosis, can happen to anybody, anytime. Now I want to give you a sobering thing to think about. Don't want to scare anybody. Every one of us in here, and I'm going to include myself because I'm part of this class too, okay? Every one of us in here produces a very tiny, small number of sex cells that have the wrong number of chromosomes in them. Eggs and sperm that are either missing a chromosome or have an extra chromosome. Everybody. And so when you go to produce a child, you literally kind of hope and pray that it's a good egg and a good sperm that get together. Now the odds are with you. They're in your favor, okay? But it's a mistake in meiosis. It can happen to anybody, anytime. Doesn't run in families at all. Bless you. Back to what we talked about a second ago. This year in Indiana, there will be, let's say, 350 to 500 babies born with trisomy 21. Anybody want to take a guess as to how many babies are going to be born with monosomy 21? Where they only have one chromosome 21?
Around the same.
How many?
Around the same.
You would think around the same.
Is it worse? Is it not born?
0.
Because they won't survive.
They don't survive. 0. If a fertilized egg ends up with monosomy 21, only one chromosome 21, it'll die. That fertilized egg will die very early, like in the first week or two. And it could be early enough, so early that mom wouldn't even know she conceived. So it looks like if you're missing chromosomes, that's more harmful to the development of the embryo than if the embryo has extra chromosomes. And I guess that kind of makes sense. Does that make sense? That if an embryo is missing chromosomes, that it would mess things up more?
It makes sense if you think of it like construction. If you don't have enough stuff, you're not going to be able to complete the whole project. But if you have more than enough stuff, you just, maybe you can like build a little more later.
Excellent. You're missing information, right? Yeah. Now here's the thing that a lot of people don't realize. Here's a question that sometimes people will ask. Well, I haven't heard anybody in the halls of Jefferson High School recently ask this question. But I have heard people ask this. How does an extra chromosome cause a birth defect? I mean, so a baby has an extra chromosome 21. How does that cause a birth defect? I'll show you. I just happen to have chromosomes in here. Now let's pretend that this is a chromosome before it duplicates itself during interphase. Long string of genes. Okay, let's pretend that each bead is a gene. Okay? Actually, this is still an oversimplified model. Why would this be oversimplified? Well,
Because those specific genes or clumps have spread out.
okay, right. It's not quite this simple. Right. And they're kind of spread out. They're not, they're in clumps, some of them. Also, chromosome 21 alone has close to 500 genes along its entire length. So this isn't quite 500. Isaac, you were going to say something.
No, I wasn't.
What? Somebody say something? Oh. So look at these two chromosome 21s. Okay, look, see them? These are the two chromosome 21s. Oh, stop that. Stop that. Get a nucleus. Okay. These are the two chromosomes. I could never tell jokes.
We went out.
Two chromosome 21s. Okay. Let's say we've got somewhere between 350 and 500 genes along the entire length. Can you see that if you get 3 of these, you're not getting an extra dose? You're not just getting an extra chromosome, you're getting an extra dose of like 300 to 500 genes. See why that's a problem? And so you're throwing all these genes out of balance. A child with trisomy 21 doesn't just have an extra chromosome. He's got an extra copy of 350 to 500 genes, and so that's why it's a huge deal. Make sense? Sort of. See, our genes are supposed to work in this delicate, it's called a duplicate balance. Now, how does this work? Nondisjunction. Quick little review of meiosis. Quinn, does this look familiar? Painfully familiar? Okay, let's say that this is our red chromosome. Now, the yellow doesn't show up, I've noticed, so I think I'll use
Yellow.
orange. We'll call that yellow. So there's a pair of chromosomes. Let's call that chromosome pair number 21. And we're looking at them before they've duplicated. So they duplicate. Here we go. Then they look like this. Okay. So now we're looking at chromosome pair 21, like this, okay? Oh, and when they snuggle together like that, actually, what's that called? When the chromosome pair does this during meiosis. Begins with a T, ends in ad.
Tetrad.
Thank you, Ashley.
Oh.
Yeah.
I really call him, oh, sorry.
Tetrad. Cole, you get this one. When they're together like this, what do?
They cross out.
They what?
They cross over.
Excellent. Thank you, Cole. Good answer. Good answer. Because I'm with it on a Friday afternoon. Okay, they cross over. Okay, now, I'm not going to draw in the crossing over right now, if that's okay with you. Let's do meiosis 1. Tetrad separates like this, and so let's put the red chromosome in this cell, and we'll put the yellow chromosome in this one. So meiosis 1 happened without a hitch. Thank goodness. Now we're going to do meiosis 2. Watch the two red chromatids pull apart. There we go. Now let's do meiosis 2 for the yellow chromosome. But let's say, just an accident now, nature is good, but it's not perfect. Things go wrong sometimes. Hurricanes happen, tornadoes happen, earthquakes happen, and sometimes chromosomes forget to pull apart during meiosis. So watch this. Maybe the two yellow chromatids are just a little slow pulling apart. The cell pinches in two, and they both get trapped in the same sex cell there, like that. That's nondisjunction. Make sense? That's just the failure of chromosomes to separate during meiosis. Now, if this meiosis happened in a guy, let's say, and these are sperms now, can you see what's wrong with that sperm right there? Yeah, it's got two 21s in it. Sperm has two chromosome 21s in it. If it fertilizes a normal egg with 121, you get what? Trisomy 21, right? Down syndrome. Makes sense?
And what happens if the egg has no 21s?
Oh, good question. What would happen if the egg happened to undergo nondisjunction? You had an abnormal egg that didn't have a 21 in it. And a sperm with two 21s happens to show up.
It's one of those.
I imagine the odds of that happening are astronomically
That's crazy. You think it's happening?
astronomical. It's possible that it's happened, but you wouldn't probably know. The kid would be okay.
That's crazy.
The kid would be okay.
Now, if somehow you're able to notice it's happening, like you're able to see the chromosomes happening, are you able to see there's a trisomy 21 in the embryo? Can you remove that actually?
Oh, excellent question. First off, I need to say something in addition to the kid who, let's say, gets both 21s from dad.
Right.
That kid should probably go to Las Vegas, right?
Yeah.
Really lucky. The kid would be okay, but not perfect. Because both the 21s came from one parent, the kid would be okay, but not perfect. There might be some little problems that have to do with epigenetics, which we'll get into next semester. So, but the kid would be okay otherwise. Not perfect, but who's perfect anyway? Isaac?
We're going to ask, would the child greatly resemble their dad, or would that not be affected at all?
That's a good question, and the answer is no, because there aren't that many genes on chromosome 21 that have absolutely anything at all to do with appearance. That's a good question. Good question. All right, now, back to a question that Caden asked. Did you ask about, is there any way to
Cure it.
cure it?
So if you like realize the mistake after fertilization.
You'd have to somehow turn off the extra chromosome 21, right?
Or maybe extract it.
Yeah, now here's the problem. Once a baby arrives in the world who has trisomy 21,
All of the cells have trisomy.
every cell in his body or her body has trisomy 21. And you'd have to go in somehow and turn off the extra chromosome 21 in each cell in the body, and
Aren't there trillions of cells?
trillions of cells.
Now, what if you cut out an embryo?
Oh, good question. What if you caught it when it was a fertilized egg in a like in vitro fertilization petri dish in the lab? When it's a fertilized egg, if there was some way you could scan it without destroying it and see that it has 3 21s and inactivate the extra chromosome 21, then that embryo would be okay. So in the future it may be possible. Not today. Not today. Now, it's kind of interesting. Remember bar bodies? I hope you say yes. Remember bar?
Yes.
The reason the extra X chromosome in the female turns off and becomes inactive is there's a gene on the X chromosome. This is free of charge. There's a gene on the X chromosome called exist. X-I-S-T. Exist. And that gene on the X chromosome is what turns off one of the two X chromosomes. Turns it off.
What if we could do that, like remove it?
Well, I was reading an article just yesterday. Some researchers, now this was in the lab in a petri dish. They were growing in a petri dish human trisomy 21 cells in a petri dish. Now these weren't embryo cells. These were, I can't remember, I think they were skin cells. But these were skin cells in a petri dish that were trisomy 21. And they managed to insert that exist gene, which is on the X chromosome,
Exactly.
into the petri dish, and it turned off the extra 21 in those trisomy 21 cells in the petri dish. They turned it off. And then those cells became more normal in terms of how fast they were growing and how healthy they were. That's kind of cool. Now, we're a long, long ways from curing trisomy 21 in people, but that's like an early first step. They figured out in a petri dish how to shut down that third chromosome 21. I thought that's pretty interesting. The thing about human genetics that is fascinating to me is new things are being discovered all the time. Genetics is the most rapidly developing field in all of biology. And all of medicine now is becoming based on genetics. All of medicine. So it's an important field. I mean, and new things are being discovered all the time. And it's a challenge for me to keep up. But I love that challenge. I couldn't imagine trying to teach something like Latin. It doesn't change much, you know. Yeah. I remember when I was in high school, we had an English teacher, and she taught, well, obviously she taught senior English, but she also taught Latin. And I said in class to her one day, I probably shouldn't have said this, I was kind of joking, and I said, Latin is a dead language. Ooh. She tore into me. Now I can understand now why. I look back and I feel guilty for saying that. I was just trying to get her riled up a little bit.
Oh, you are? Yeah.
And I said, oh, Latin's a dead language.
Yeah, but she finishes it.
She got mad at me, you know.
Isn't it?
Huh?
But it is.
Sort of, yeah. All right. So, nondisjunction. Now, this is probably the most well-known example of a result of nondisjunction, right? Down syndrome. And I can't remember where I saw this, but I thought it was pretty cool. Friends don't count chromosomes. You got 4 teenagers there who have Down syndrome, and I thought that was pretty cool. Oh, here's a little girl with Down syndrome. Now, when people have Down syndrome, trisomy 21, when they're very, very young, sometimes it's hard to tell. Okay? But as they get older, the characteristics become a little more obvious. Does anybody know anybody who has trisomy 21? Okay. Relative? Anybody have a relative? I have a girl in first period genetics. Her brother has Down syndrome. And so I tried to make it clear, and I hope it's clear. It doesn't run in families normally. 90, 97% of the time, it doesn't run in families.
Do you know if autism runs in families?
We don't know much about autism.
I think they're like studies on
Yeah, they're working on it. If autism, if genetics has anything to do with autism, it's surely polygenic. In other words, it's not one gene that's messed up, but several genes controlling personality and intelligence and behavior, all working together in ways that we don't understand. Whole constellations of genes in combination with environmental factors, big mystery.
it. I always thought it did because our family has a very, very long history of autism. Just so many people in our family.
If it is polygenic, then that would be expected. With polygenic traits, and we'll get into polygenetic traits next semester, polygenic. But with polygenic traits, you can't predict, like with percent risks like you can with single gene traits. You can't predict the odds of the conditions showing up in the children or in the grandchildren like you can with single gene traits. All you can say is it tends to run in families. That's all you can say. You can't predict. But like with single gene traits, remember, if mom's a carrier, let's say Tay-Sachs disease, and dad's a carrier of Tay-Sachs disease, what's the probability of the kid having Tay-Sachs disease?
25%, 50% carrier.
25%, right? Remember that stuff? Okay, with single gene traits, you can predict those probabilities, those recurrence risks. With polygenic traits, you can't. All you can say is they tend to run in families. So sorry, that's not the clearest answer you were looking
No,
for.
that's
But
not.
that's a big, autism is still a big mystery as far as how much we know about it. So anyway, we're going to find out. 97% of the time, chromosome disorders like Down syndrome do not run in families. They're just one-time random mistakes in meiosis, nondisjunction. Okay? Speaking of Down syndrome, in our church we had a preacher who he left a couple of years ago and took another job out in Kansas, another preaching job. And he and his wife have 2 little boys, and then they adopted a baby girl right before they left, right before he got this other job out in Kansas. They adopted a baby girl, and they, on purpose, knowingly, adopted a baby girl who has Down syndrome. And so, I remember some people thought, wow, why would you do that? Well, that would be a lot of extra heartache. I mean, it's not easy having a child with Down syndrome. A lot of them do pretty well, but it's still not easy. And then, so they adopted this baby girl, and then this job opportunity became available for him, and so they moved out to Kansas. And after they moved out there, they found out that where they lived in Kansas City, there's a hospital that is like the leading hospital in the world in dealing with children with chromosome disorders. So it was just kind of it worked out, it worked out for them. What are some characteristics of Down syndrome? You guys know?
Can't learn.
Right. The obvious one is mental handicaps. But there's another one. Half the kids with Down syndrome have heart defects of some kind. And that's a real serious issue. So whenever a baby's born with Down syndrome, they always, one of the very first things they do is check the heart to make sure there are no heart defects. Okay. There's her karyotype right there. Trisomy 21. Right there. Aneuploidy, right? Okay, here's what we're going to do. if you'll take your study guide, we're going to jump a little bit here. We're going to jump ahead. Take your study guide and jump all the way over to page 9. And then we're going to come back and fill in the intervening pages on Monday. Jump over to page 9. See where it says at this time go through the computer lesson entitled Examples of Chromosome Problems. And start filling in pages 9 through the top of page 17. So we're going to go back to the computer tutorial on cytogenetics. And you're going to click on the lesson called Examples of Chromosome Problems. And that's where you're going to get introduced to several different kinds of chromosome disorders in addition to Down syndrome. Okay?